This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 214, recorded on January 4th, 2013. Time to get used to a new year. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent. Belated happy birthday in person.
Thank you very much. It happens once a year.
Yep. We exchanged emails, but now I get to say it.
Thanks to everyone on Facebook also who wished me happy birthday.
Yeah, something like 3,000 likes. Wow.
You know, it's a lot of people. I never had so many people wishing me happy birthday, so that's really cool. Thank you all. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you today?
Doing okay. Weather is finally not quite as frigidly cold as it was.
What do you got there?
We've got 3 degrees Celsius, 37 Fahrenheit today, which it's the first time it's been above freezing in about 4 or 5 days, I think.
Clear skies?
Yeah, nice. And actually, it's clouding over a little bit now.
How about you, Kathy?
25 Fahrenheit, -4 Celsius, and bright sunny day. It's nice.
-4 Celsius. Oh, pretty cold there.
Yeah, but it's sunny.
It's normal winter in Ann Arbor, huh?
Yeah, we're happy to have sun.
Here it's 2 degrees Celsius. It's sunny skies. Very nice day today. It was colder earlier in the week. That's right. Well, we are in the new year, 2013. Got to start getting used to writing it everywhere. And the first episode of TWiV for the new year. And we have lots of follow-up today. I wonder why I have so much follow-up on the last couple of episodes.
We must have screwed up a lot of stuff.
Or people just have a lot more time over the holidays to listen and get caught up.
Yes, that's true.
That could be it. I just copied a couple of comments from Facebook. One from Deborah about 123. No, can't be 123. 213. See, I'm dyslexic. I didn't write it. 213. One of the best podcasts ever. I so wish I had some kind of college degree. Love you all and thank you. Well, you don't need a college degree to listen to TWiV, but I guess if you wanted to do some science, you probably might. But Thanks for listening. Danny, also on Facebook, concerning the ignorance episode. This was one of the best gifts I've been given all year. The interview is fascinating, touching, and gives me hope. I'll most definitely be reading the book now. Stuart Firestein's combination of idealism and creative strategic thinking is a terrific example of the strength found in combining arts and sciences. So check out Ignorance. The book is really good, too.
Yeah,
it's a great episode.
Did you read the book, either of you?
Not yet.
You should.
I did. I loved the book. I thought it was brilliant.
I gave it to my lab for Christmas, or the holidays, I should say. I gave them all a hard copy, because I wanted to give them something. Mm-hmm. I'd never given my lab anything in 30 years.
I know.
So you gave them ignorance?
I gave them ignorance, yeah.
You gave them ignorance.
Nice. Sorry, Alan.
That's okay.
Yeah.
I managed to pick up ignorance on my own.
No, I mean, back when you were in my lab, I never gave anyone anything.
Oh, that's fine.
But this really struck a chord, and I thought people in the lab should have it. So there you go. The next one's from a guy named Rich. Dear Twix gang, Rich stumbled and fell again in TWiV 213 over polyomavirus nomenclature. You would think that after all these years, he'd get it straight. Brachoviruses and ichnoviruses refer to 2 different lineages of polyomaviruses. They both package multiple circular double-stranded DNAs and have similar life cycles. However, they inhabit different families of wasp. They have different capsids and modes of egress, and they package and deliver different host genes. These facts suggest independent evolution. Nudibvirus refers to a modern double-stranded DNA containing insect virus that is most like the virus that probably integrated into the wasp genomes long ago to provide the capsid and transcription genes that contribute to polyomavirus replication. Get it right next time. I love the show. Of course, that's Rich Condit. I should imitate his voice, but I can't. Love the show. Well, that's not it. Next one is from Trudy. Hi, Twivers. I'm writing this email in response to Rich's comment about nudie in episode 213. My prep notes for this episode, since I was a guest on that particular episode 179, I made the following summary to help me understand the relationship between brachoviruses and nudie. The hypothesis is that the viral origin of these brachoviruses goes back to nudie, which are large dsDNA viruses that are closely related to the baculoviridae, and which often establish persistent infections in arthropods. In this paper, they did a deep sequencing analysis of both viral and wasp genes during replication of brachoviruses to address the question of whether other nudie-like genes exist or whether the genes identified to date are present in all brachovirus-carrying wasps. I hope this helps, and I really didn't mean to insinuate that Alan is short. He just sounds really tall. Happy New Year, Trudy. Thank you, Trudy.
I'm just shorter than you thought I would be.
Alan, why don't you take the next one?
Sure. Winkler writes, Dear TWiV panelists, I never miss an entire podcast. Thanks. Please allow me to expand on Alan's point that the human genome is now being examined to predict idiosyncratic drug reactions as it is to understand viral susceptibility and virulence. I played a small part in the very first use of pharmacogenomics in clinical medicine. This was a multi-center, multinational effort spearheaded by GSK, GlaxoSmithKline, the company that previously started modern antiviral therapy, acyclovir, and antiretroviral therapy, AZT. We found that subjects who have the B*5701 HLA marker might experience a fatal allergic reaction when exposed to the antiretroviral drug abacavir. HLA B*5701 negative individuals are not at risk for this side effect. Now all patients get this blood test before abacavir is prescribed. Thanks again for keeping me so informed about the rapid evolution of virology.
It's very cool.
Yeah.
I guess the plan is eventually you will profile everyone and before give them anything, right? You have their genome sequence, you'll have their transcriptome, their proteome, and the doctor will look in the file and say, oh, you can't have this drug or you can't have that one, right?
Depending on the drug. Yeah.
It's cool.
Yeah, of course, this also brings up lots of logistical issues, but eventually when the genome sequencing is cheap enough, everybody will just have that done at birth. You'll just have that as part of your medical record.
Yep, exactly. Kathy, can you take the next one?
Robin writes, educated by TWiV. As mentioned by Dr. Firestein, teaching is lighting fires rather than filling buckets. Well, a small fire was lit in this retired emergency physician. Thanks to TWiV, I looked up Wikipedia and discovered that placental mammals appeared before the extinction of dinosaurs 75 million years ago, according to paleontology, and up to 100 million years ago, according to molecular phylogenetics. I appreciate TWiV for all it does.
Good to know that Robin, who writes often, is a emergency doc.
Yeah.
Interesting. And retired at that.
Mm-hmm.
The next one's from Howard, who writes, Hi, Vincent. During a recent conversation with Stuart Firestein, he mentioned the first president of Israel, Chaim Weizmann, had been trained as a chemist but had not worked as one. Although better remembered now as one of the early proponents of the 20th century Zionist movement to create a homeland for the Jewish people, it was his brilliance as a scientist that prepared the way for his passion to become a reality. Weizmann was a pioneer of using large-scale fermentation to create industrial chemicals. He developed the ABE process using Clostridium acetobutylicum to produce acetone, butanol, and ethanol, 3:6:1 ratio, from anaerobic fermentation of starch. Weizmann's adopted nation of the United Kingdom had a munitions problem during World War I because the main supplier of acetone used to make the explosive cordite had been Germany. Several British breweries were commandeered to use Weizmann's ABE process, which enabled the UK to continue munitions manufacture. The acknowledgement of this and a couple of additional chemistry-based achievements led to the next steps. Weizmann had known former UK Prime Minister Balfour for quite a while and is believed to have been crucial in developing the Balfour Declaration of 1917, promising creation of a homeland for Zionists in Palestine. The Weizmann Prince Faisal agreement in post-war negotiations was also helpful when Faisal became the first king of the newly created country of Iraq. The rest of the story is too well known to go into, but although he was honored as Israel's first president for his political efforts, these were made possible because of his scientific contributions. There is a current ironic twist to the story. Although it's been said that God gave Israel everything but oil, modern versions of Weizmann's ABE process may make enormous petroleum reserves less valuable. The primary product of the process is butanol, which can be used as a direct replacement for gasoline as a transportation fuel, and unlike ethanol, has equivalent power density and can be shipped by pipeline. The process can be carbon neutral, but production still needs to be made more efficient, which several university and commercial labs are doing. This may be an example for Dr. Firestein's hope for more basic science, since when Bayer Inc. first isolated this anaerobic bacterium, he was just describing the range of environmental microorganisms that were not involved in disease processes and was probably amazed at its eventual use. Another topic, I'm looking forward to being in the audience at the ASM General Meeting in Denver this year for a recording of a new episode of one of your wonderful series. Best regards, Howard. It's a cool story, which I did not know. And I sent it to Stuart, and he said, wow, I didn't know that. So thank you, Howard. Alan, can you take Jenny's?
Sure. Jenny writes, Dear TWiV, I was very interested in your thoughtful discussion of Chadwick's letter in episode 213 and the challenges of balancing a science career with raising a family. As a person raised by two scientists, I thought you might be interested in what it is like from this side of the bench. My father was a doctoral student in biology when I was born, and my brother was born during Dad's first year as a postdoc. My mother went back to school for her doctorate in molecular biology when my brother and I were in elementary school, and I was in high school when she received her degree. My parents both worked full-time while I was growing up, but they were always home for dinner. We had family time in the evenings and on weekends, and I know my parents frequently brought work home to do after they put us to bed. I'm embarrassed to say I once selfishly accused my mother of not being a normal mom like the stay-at-home moms of my friends, but I now appreciate that she was an incredible role model for me. It cannot have been easy to be a female doctoral student with kids during the 1970s and 80s, especially in a field like molecular biology. But my mother's perseverance and success taught me that I could pursue my own challenging career and succeed. What is it like to be the child of scientists? In a word, cool. My parents' labs were home to us. I loved going to work with my dad on the weekends. He would set us up at a bench with a roll of brown paper and some crayons and let us go to town while he and his students worked around us. His students became part of our family, and we loved the cookouts and lab parties and journal club pizza nights at our house. My natural curiosity was constantly encouraged and reinforced by everyone in my parents' science circles. And when it was time for science experiments for school, I had access to the kind of equipment that made it possible for me to find the LD50 for bleach in a minnow tank. And an unlucky grad student even helped clean up the mess. No one in my school could match that. Dinner table conversation at our house was, and still is, about endangered species, electron microscopy, climate change, gels, grant proposals, Cold Spring Harbor, manuscript submissions, and the agonies of academic life. Sometimes it was hard, especially when my dad was away in the field, but I treasure the memories and excellent stories of being a scientist's kid. Most kids didn't have salamanders in their fridge or a crocodile head in the freezer. Most kids didn't earn money by writing out reprint cards for Dad's sophisticated index card filing system, 5 cents a card. Most kids couldn't spell schistosomiasis, let alone know what it was. Finding success while raising a family is a challenge for many professionals, including scientists. There were definitely occasions when the lab came first, and that was hard for me to understand at the time. But my parents shared their science lives with my brother and me in ways that shaped who we are and how we think. I consider this to be a tremendous gift. My only regret is that I never asked my mom to show me how to do a plaque assay before she left the lab for freelance science writing. After listening to 213 TWiVs, I realized that would have been cool. Cheers, Jenny.
Great letter, huh?
Yeah.
That's great. Yeah.
What is the LD50 for bleach in a minnow tank, Jenny?
Wow.
Jenny, I met Jenny a while last year, 2011, at a meeting. And I didn't know this. You never told me, Jenny. It's cool. Well, it sounds like you lived close to the lab, and that helps a lot. Well, I stayed home for my birthday on Wednesday, and I got to see my kids coming home from school, and we had dinner together. How's that?
Excellent.
I feel very guilty about that, about not having done it more, but oh well. They don't seem to mind.
Yeah.
Kathy, can you take the next one?
Sure. Carolyn writes, I just wanted to thank you both for featuring our placenta work on TWiV. I awoke to an inbox full of emails from folks who had listened to the most recent podcast, as I intend to do today. I assume you have heard this many times, but I just wanted to reiterate what a really fantastic influence TWiV has had on our graduate students, microbiology and molecular virology here at Pitt. By listening to TWiV, our students have been introduced to new areas of microbiology and science that they otherwise would not have been. Because of TWiV, you are directly influencing an entirely new generation of scientists, which is really quite powerful. Thank you again, Carolyn. So this is Carolyn Coyne, whose work we talked about in the roundup last week, and also was on the TWiV from ASV in Madison this summer. So
Cool.
yeah.
How about over in your department, Kathy? Did students listen to TWiV?
We have some very diligent ones that listen to TWiV, and then others that don't. I just interviewed someone, an MSTP student, so that's a MD, PhD student, and I happened to mention something about TWiV, and she said, I love TWiV. I have two things on my bookmarks bar, and one of them is TWiV.
Yeah.
So, and she had missed seeing the one that you did here a little over a year ago, and she really regretted that. She didn't know about it. But so, cool. Yeah.
Okay, that's cool. I think you're right. Maybe people had time during the holidays and wrote. But we do like to hear, and as I said, if you write right after the episode, in that week you get to be follow-up, also known as FU. And we'll read your email right away as opposed to waiting a long time. Now we're going to get more follow-up, right?
That's right.
That's okay.
Well, as long as most of it's positive and not like that stuff that rich guy said.
Yeah, Rich Condit, so critical.
So critical of the
Get
show.
it, you said get it right next time.
Yeah.
Wow. Okay,
Maybe
will.
we'll just have to get him on the show.
It's not a bad idea. All right, we have two papers today to start off the year. The first is a paper in Science, actually sent to me by Saul Silverstein, or Silverstein. I don't know what his mother prefers. Silverstein? Yeah, he says Silverstein. You know, we have a Sam Silverstein here, too.
Mm-hmm.
It's confusing. And this is the title of this is Coagulation Factor X activates innate immunity to human species C adenovirus. It's a science paper with a slew of authors.
Right, so it's a team effort. The senior authors are Phoebe Stewart, who's a cryo-EM expert, and Dmitry Shakhmedov, who's been very involved in adenovirus entry and innate immunity. So,
this is quite a compact paper.
Yes.
Yeah.
That's a nice way of putting it.
Every sentence is like a normal paragraph in another journal. It's very constricted here.
An enormous amount of work condensed into I think an absurdly small space.
Yeah.
I think it's to the detriment of understanding the work, really.
Yeah. It really, there are a lot of people who would probably get a lot out of this paper if it were allowed to occupy the space that it needs.
Right. And
so we can just jump to my comment
Sure.
Bruce Alberts has an editorial in Today's Science, January 4th, where he describes a new format for some science papers where it sounds like they're going to have some kind of expanded abstract, and the paper itself will be in the print copy of the table of contents, but in the paper version of the journal, it won't be the whole copy. But then online, it'll have everything. And what he says about it is, let's see, this will be good for papers that contain numerous micrographs that require high-resolution displays, or others that require more space due to a complexity of analysis, which I think is really the case for today's paper. Yeah. It's just really complex, and there are whole sections where there's just a sentence and there's not even room to explain why they did that experiment.
So they're introducing a new format in which the papers will be partially redacted.
Really?
No, they're going to be totally available online.
The online version will be totally available.
Right, and the mobile versions and all of that.
Well, totally available behind the usual paywalls.
Yeah.
Right.
I mean, this should not be a criticism of the authors, of course, because
No,
I'm sure if they had been able to, they would have written a very, very long paper. But it's impossible. We teach students to write a paper by explaining what the question is and how you did it and give the results. And, of course, this goes contrary to everything that we teach. So I don't know why science has to perpetuate that. I'm complaining. But also, there is so much supplemental material here. Mm-hmm. In parts, every sentence has a reference to a supplemental figure, which then means you have to go to a separate file and look it up. I just think that's ridiculous. That is done that way for the convenience of the journal, and it does not help convey the science itself.
Yeah, it's done because there's limited page space in the print edition. And if you're a journal that's still heavily invested in the print world, like Science or Nature, you have to keep enforcing these limits, which, this made sense back when you could publish a paper with 3 figures worth of data.
Yep.
But this no longer makes sense now that you're required to have 5 or 6 doctoral theses worth of work to publish a single paper.
Yeah. Well, if I were editor of Science, I would change that.
Yeah. Maybe I think this new format will go a long way toward that.
It helps.
Yeah.
If you remember, the eLife paper we did on the Hep B receptor was just wonderful. It was totally comprehensible, although there was a lot of data there, pages of pages, but it was there. I don't think we had to go to supplementary data very often.
Right.
That's the way it should be.
Right.
All right, so publish your papers in eLife.
Right.
This paper deals with adenovirus. Kathy, why do they say human species C? Is that...
So there are now, species A through F, or maybe G. They used to be called groups or subgroups, and then they changed the nomenclature to call them species.
I see.
So,
and they're all human strains, right? A through whatever, or
A through F or G, right?
And they're distinguished by sequence primarily, I suppose, right?
Right.
Okay. So this deals with human species C adenovirus, and their interaction with coagulation system, which is not something I think we have ever mentioned here on TWiV, because it has to deals with in us anyway, blood clotting. But it turns out that it's a very ancient immune system. In fact, their first sentence in this article, in the abstract, is that the coagulation factors play a host defense for horseshoe crabs, which of course are very ancient animals. So I went and looked it up, and indeed, when these crabs are, say, infected by a bacterium, the coagulation system kicks in. It makes a gel around the bacteria. It helps to digest them by bringing in various enzymes. Really neat. So in this paper, they ask, well, is there some role for this coagulation system in immunity in us, for example, in mammals? And I suppose that previously it had been found that adenoviruses bind what's called coagulation factor X.
Yes, except it's factor 10.
Factor 10?
Yes.
The coagulation factors are Roman numerals.
Right.
Oh, Mac OS 10 and Mac OS X, right? The same thing.
Right.
I like FX because, special effects.
Yeah.
Right.
And the X factor is a show, right?
Right.
What's that about? Coagulation proteins?
They have a reality show competition to see who can clot fastest or something.
That would be a good show, I think.
Yeah, maybe. Be a short season.
All right, let's see if I can remember. F10, I have to call it?
Factor 10, yeah.
Factor 10?
Yep.
All right, well, I'm going to probably go to
You've probably heard of factor 8. So that's another one of the factors.
Yeah, factor 8.
I remember having to memorize these for a test in college at some point. It was dreadfully boring because, it's just this factor does that cleaves this, and this is this whole series of cleavage events that occur that produce a clot.
Right.
But this is much more interesting.
It's very interesting. So apparently activation of coagulation factors can trigger inflammation. So they asked, is there some role of the interaction of this factor 10, or also known as FX, with adenovirus in inflammation? And the first thing that they do is to solve the structure of factor 10 bound to adenovirus by cryo-electron microscopy. That's where Phoebe Stewart comes in. And that's the first figure, and it's just a beautiful figure. Of course, adenovirus structure is beautiful, has those wonderful pentons and hexons. And then with the added factor 10, that factor 10 is binding to all the hexons on the surface. So they have actually factor 10 in red, so you can really beautifully see it binding to those hexons. Binds to the center of each hexon. They have a wonderful blow-up of one hexon in one part of the figure. And there are actually two movies in the supplementary data, which the first one is an image of the whole virion rotating and showing the absence or presence of factor 10. And then I think the other one is a close-up view of the factor 10 bound to the hexon and how it interacts with a specific residue or residues of hexon. They're pretty cool. Those are worth looking at, but you have to probably be a subscriber to see those, unfortunately. So the structure allows them to see what residues interact, which hexon residues are interacting with factor 10, and they identify, 1, 2, 3, lysine. See, factor 10 residue K10 and hexon residues, glutamic acid 424 and threonine 425. And that's important because, as you might guess, the next step is they change the hexon residues to see what they do to factor 10 binding. And in fact, they show that one mutant, what is it, TEA, I forgot what that is now.
Well, that's the mutation of, where they mutate residue 423, 424, and 425 to T, E, and A in those three positions.
So the third position is changed. Oh, sorry, E.
They change several of them, right? So the wild type sequence is TET. Okay. Oh, so in this one they only changed the third amino acid. Right.
Yeah. So the TEA mutant doesn't bind factor 10 at all, which is pretty cool. So a single amino acid change is enough to block that. So it's a hexon change, it blocks factor 10 binding. This virus doesn't infect CHO cells, Chinese hamster ovary cells, which apparently the presence of factor 10 is important for infection. But in other cells like A549, which is a lung epithelial cell, is that correct? Mm-hmm. The factor 10 binding mutant infects quite fine. So they have now a virus that doesn't bind factor 10, which is a very good reagent, of course. And here we've described it in 10 minutes. It probably took them a long time to get to this point. Because they had to have the structure first and say, oh, look at these residues, and make the virus. So then they say, what's the difference in the innate immune response? If you have, first they look at the innate response in adenovirus infection. They actually look at the whole transcriptome. They analyze the transcripts for 28,000 genes in in infected cells, which is just amazing that you can do that.
This is where their team science comes into play, because these four authors, the second line from the bottom on the title page, McDonald, Bamler, Bayer, and Farron, were all involved in the microarray analysis. So that very important group of people to be able to do that.
Yeah, so they have to look at all of the transcripts that are induced, say. So they look at things that are induced more than 1.5 times. Here they look in the spleen of mice 30 minutes after injecting adenovirus. And if they find, of course, that a good number of genes are upregulated, I think 519. They're split into 4 different categories. And here's where the bioinformatics people come in. They do gene ontology analysis, and they put all these genes into groups. So you could say, some are involved in stress, metabolism, apoptosis, and so forth. So you want to know what pathways are activated. And in the end, they say, well, the important pathways induced by ad infection in macrophages, because those are the cells here infected, are NF-kappa B and a couple of other pathways. But the real key here is that when you infect with the TEA mutant, you don't get upregulation of a number of cytokines and chemokines, which shows you that, or tells you that the presence of factor X is important for inducing these cytokines, and I think they included IL-1B, interleukin-1B, interleukin-6, and MIP-1 alpha, macrophage inflammatory protein 1 alpha. So you have to have factor X present in order to induce these cytokines. So these cytokines are induced in wild-type adenovirus infection, but not if you use the mutant lacking the ability to bind factor X. The induction of these pathways depends on toll-like receptor 4. They do a lot of work looking at mice lacking the various sensors, the various toll-like receptors. And it turns out that TLR4 is important for sensing adenovirus in the context of factor 10. So factor 10 has to be bound to adenovirus, and then it's recognized by TLR4, and that leads to the inflammatory induction that is typical of adenovirus infection. And so if you take knockout mice lacking TLR4, they're not good at clearing the virus, and it replicates and overwhelms the animals. So that's pretty interesting that here you have a coagulation factor binding the virus, and then that is recognized by TLR4 to induce the innate response, the cytokine response. And you might ask, well, why is factor 10 recognized by TLR4? Because it's self, right? And so
right, it's a host protein.
It's a host protein. And I think the idea of the authors is that by binding to the hexon, it's adopting the symmetry of the hexon. So it's changing the structure of the protein, and that is what allows it to be recognized by TLR4. So that's very cool, and that's clearly something that you could pursue and really nail down.
Right. So they in the commentary they make the specific point that this association between the TLR4 and factor 10 can result in the formation of a pattern established by the symmetry of the virus.
Right.
And the TLR4 typically recognizes LPS, which has a repeating pattern, and so you could make conclusions about the repeating pattern from the symmetry of the virus.
Yeah. Now, I think they also mentioned that there was some other precedent for clotting factors binding to virus. I seem to remember herpes virus. Where was that?
Oh, right. I remember that as well, but I don't know where I saw that.
Oh, yeah, here we go. Direct binding of factors 8 and 10 was recently reported for herpes virus type 1, herpes simplex type 1. I missed that one. That's 2006. That's not very recent. So what I think is interesting is that, so if this is an ancient immune system of sorts, the coagulation system, clearly it's still working in mammals, right?
Yeah.
It's still there for AD, and even though AD is sensed also by other ways, not involving factor 10, this pathway is still there, so maybe it's still important, right?
Mm-hmm.
Yeah.
And it may apply to other viruses as well.
The other thing that's interesting is that there are some adenoviruses that don't interact with the factor 10 or factor 9,
Right.
another factor. And then in the case of mouse adenovirus, there's a report from a Belgian group that it does associate with human factor 10 and with murine factor 10 and 9, but that doesn't increase its cellular attachment to hepatocytes.
Mm-hmm.
So there's a whole spectrum of maybe interacting with factor 10 or not, and maybe that brings you to a different cell type or not, and it's just kind of, I don't know, fascinating to think about the evolution of this.
So mouse AD does not interact with factor 10, you said?
It does interact with factor 10, but that interaction does not increase its cellular attachment to hepatocytes.
Okay, as it does for the human C strains here, yeah.
Right.
Right, so depending on the host and the context and how the binding occurs, the virus might be able to evolve some kind of tailored targeting.
Exactly.
Yeah. Kathy, do you think that factor 10 binding to mouse AD is involved in sensing?
I have no idea. It could well be. We haven't done any experiments, nor do I know of anyone else who has to look at that. It's an intriguing possibility.
Yeah.
Yeah.
Well, if it's binding, then it's probably being sensed, I would guess.
Right.
If it says, they say, adopting the conformation of the hexon and so forth.
Right.
Yeah, and I think in the commentary, the authors also pointed out that this is kind of an interesting highlight on the evolution of adaptive immunity. That you've got this system here that is you've got a factor binding to a pathogen, and that allows the immune system to recognize it,
right,
which is kind of a precursor concept of what antibodies do, but it's not as specific.
Right, opsonization, right?
Right.
Mm-hmm.
Yeah. It's funny, we just did a paper on TWiV where they talk about opsonization of bacteria by antibodies. And just yesterday, someone sent an email and said, is there any analogous thing in viruses?
Yeah.
And here you go. Maybe this coagulation is similar. Because I don't know of any... Well, I guess there's some examples where antibodies bind viruses and help them get into macrophages, like dengue. That's thought to be the way that you get that serious dengue on the second infection.
Right.
But for bacteria, opsonization is a way for antibodies to lead to uptake of the bacteria into macrophages, so they get rid of them. So I don't know of any virus antibody interaction that leads to that. Do you? Does anyone?
Well, I guess I'm thinking about the complement opsonization of some envelope viruses like herpes and so on. So isn't that
opsonization or lysis? Do you mean?
I guess I just mean opsonization.
Yeah. Maybe that's right. Yeah, I have to look in that. That could be well. So that stimulates uptake of the larger viruses. Yeah, that could be. So this is cool. I thought this was a neat little paper. It's short, but shouldn't be so short.
Yeah.
Here, one more thing I wanted to bring up here. They say that the use of adenovirus vectors for gene therapy in humans has fallen out of favor over the past 10 years. Of course, everyone probably knows that a patient died upon getting adenoviruses intravenously, or he says hepatic delivery, but I guess that was intravenous. Mm-hmm. And that's partly because of this inflammatory response that ensues when adeno triggers the innate response. Mm-hmm. And so they conclude by saying, although ad vectors are excellent models to study antiviral innate immunity, their de-immunization for treatment of genetic disease likely remains a challenge. What do you think about that, Kathy? Is that a fair assessment?
There's a lot of ways that people are trying to do that.
Yeah.
I think it's true. It does remain a challenge. But people are creative and are thinking about lots of ways to do that.
Yeah.
Yeah, and it's not like that's the only hard problem in gene therapy.
No.
So there are a lot of people who work in gene therapy are accustomed to tackling pretty big stuff.
Yeah.
You can put a lot of genes into an ad vector. That's the nice part about it, right? Mm-hmm. And other vectors are kind of limited. So
yeah, there are actually a lot of characteristics of adenovirus that are attractive for this, and other than the inflammatory mechanism, it's not generally viewed as a horrifically harmful virus. They are fairly prevalent, and most people survive them. And you've got a lot of space to put stuff in, and it's a DNA virus and pretty easy to work with, and all that.
All right.
I was just going to mention that we've been talking about this commentary. It's in the perspective section of Science, and it's two other authors, Roland Herzog and David Ostroff, who are evidently at University of Florida, but not in Rich's department.
Oh.
Mm-hmm. Is that probably is behind the paywall also, right? This commentary.
I think so.
I would guess.
Yeah. Pretty much most of Science is behind the paywall.
Hmm. All right. The second paper was brought to my attention by the primary author, the PI, Patty Pesavento, who is a professor at UC Davis, University of California, Davis. She wrote, "As sure as we are that you get requests daily, we could not help but let you know that we've described an outbreak of brain tumors in raccoons in California. 12 total to date, all in the same place, frontal lobe, all within 100 square kilometers. And here's the interesting bit. 100% associated with a novel, and we suggest oncogenic polyomavirus that we've called TWiV. Interesting complement to your interview with Patrick Moore. End host, species jumping, maybe, and yes to those questions. So we'll come back to those questions. So that's where I came across this, and then it got published, and I said, we should do this. It's a pretty cool story.
Mm-hmm.
Yeah.
I didn't know that tumors were rare in raccoons.
I have to say I cringed when I saw that sentence because I thought, oh no, there's going to be a whole woo-woo industry now dedicated like the shark cartilage nonsense. But but at least raccoons are plentiful, so they can probably tolerate it.
So this is, as we said, from Davis, De La Cruz, Gianniti, Lee, Woods, Del Valle, Eric Delwart, the sequencing guy who has been on TWiV, and Patty Pesavento. And apparently, veterinary diagnostic labs receive large numbers of raccoons and carcasses. They're dead. And they want to know if they have rabies and other things. But they say in this paper, tumors of any type are rarely reported. So if you don't want to get cancer, you should be a raccoon.
Well, actually, after reading that, I thought, well, wait a second. What do we know about raccoons? They live maybe 2 or 3 years in the wild. They get hit by cars. They get rabies. They get hunted and turned into caps and all this sort of stuff. So it may just be that the odds of surviving I suspect for a lot of wild animals this is the case. They don't live long enough to get tumors.
Could be, yeah. So basically, these are animals from Northern California and Southern Oregon that they got. They're free-ranging raccoons, so they're not in zoos or anything.
They have a nomadic suburban lifestyle.
I love that.
I love it. Yes.
Mike Imperiali's lab wanted to know what free-ranging meant.
No chemicals.
All right.
Yeah.
Like free-range chickens?
Yeah, they're not.
These are cage-free raccoons.
Yeah, not domesticated and not people's pets, I guess.
And they have tumors, olfactory tract, frontal lobe, brain tumors. So they have a photograph of that. It's quite grisly.
Yeah.
Yeah.
I guess it's a section of the entire brain of the raccoon. They have a normal and then these frontal tumors.
It's really, really dramatic. I mean, I'm no pathologist, but as soon as I glanced at this figure before I even blew it up, I said, oh, raccoon B looks really, really sick.
So they looked at 52 raccoons, and 10 of them had brain tumors, so that's 19%. And they called them MPNSTs. And I had to look it up because they didn't define it. That's malignant peripheral nerve sheath tumors. Did you guys know that? You probably did.
I think you put it in show notes somewhere.
Show notes.
Yeah.
It is neurofibrosarcoma, neurosarcoma. It's a cancer of the connective tissue surrounding nerves. It's a sarcoma. And people get these too, but now we know that raccoons get them. All right, so they have these tumors, and then they decided to look for polyomaviruses because there are polyomas associated with brain tumors like JC polyomavirus, and these are glial origin tumors, which is what they found in the raccoons. So
that was my first thought was, did they go right to looking for polyomaviruses? They don't really describe that, but that's certainly the impression I got. They didn't.
It seems to be a very lucky guess from what we're about to talk about.
Mm-hmm.
Yeah, I mean, that's the way they describe it. If Patty is listening, maybe you can enlighten us. I actually invited Patty to join us, but I didn't hear from her. But I would say from the way it's written that, they thought, well, what causes glial tumors, JC polyomaviruses, so let's look for that. So they made some primers and got positive results. So then they went back in and pulled out the entire viral genomes, full-length genomes or partial genomes. From full length from 6 out of the 10 animals and partial from 4 out of 10. And when they sequenced them, it looks like a typical polyomavirus. These are, of course, small double-stranded DNA containing viruses. The genome is circular. We have talked about them a few times. And this is actually something Rich Condit used to work on. Mm-hmm. So it's too bad he's not here. But we did have an episode with Pat Moore the discoverer of Merkel polyomavirus, TWiV 160. So check that out. And it has the normal proteins that you would expect for a polyomavirus, the large T antigen, and so forth. And it seems to cluster with the Merkel. It's most closely related to the Merkel cell polyomavirus, the Moore virus, which is involved in a rare human skin cancer. So this was found in 10 out of the 19. Is that right? No, there are 10 tumors totally, I guess, that they looked at.
10 raccoons, 10 tumors.
And 10, right. And they got virus from all 10 of them, or sequence from all 10. Mm-hmm. Yeah, six full genomes and 4 partial genomes. So that's in Patty's letter. She said 100% associated with this polyomavirus, which is, by the way, is called raccoon polyomavirus, TWiV. Has nothing to do with Racaniello. The Rocky raccoon. I would have called it Rocky virus, but it's a good thing they didn't ask me. They do some work to show that the genome is episomal, so it's not integrated into the host genome. Now that's interesting. For some polyomas, integration is required for tumorigenesis because that prevents the viral replication cycle. It allows constitutive expression of T antigen and transformation. But there are other mechanisms. So the episomal state of the genome is okay in terms of its potential for transforming cells. They also show that large T antigen is expressed in the tumor tissue, but not in normal tissue of some of these animals.
It's not expressed in a whole lot of the cells in the tumor tissue. It's just kind of this
subset,
sporadic cells. Yeah.
Did you show that to Mike and get his thoughts on that?
Yeah, and that was one of his first comments was that you don't see many T antigen positive cells. So, and we both wondered why in this journal that particular figure was referred to, but was yet in an appendix. Anyway, I guess maybe because it was. Had to go search that out separately.
Yeah, that's weird. Yeah. So they believe, of course, that the tumors are caused by this virus, but of course this doesn't prove it.
Right.
So what would
they word it pretty carefully.
Yeah, sure.
But so
they believe it is. So the first question is where this virus came from, and she said in her letter, end host, species jumping, maybe and yes. So end host maybe, species jumping yes. So they must have more evidence that this has jumped from another species, I guess.
Yeah.
And what that might be, who knows? Because there are polyomas that not only infect people, but other animals as well. So it might have jumped. Mm-hmm. And maybe this is the end host. Mm-hmm. But I guess you could get some raccoons and infect them and see if they get tumors.
Yeah.
Right.
It's not an easy experiment, but I guess
it
can be done.
They shouldn't be hard to obtain. And these tumors seem to have, they believe that the tumors originated in the nose, essentially, right?
Mm-hmm. I think so.
And so I would think you could get some raccoons and experimentally just inoculate them through the nose and see what happens.
Right.
And it's worth noting these raccoons, this is not a random sampling of raccoons that show up at the lab. These are usually ones that are sent in because they bit somebody or they're suspected of having rabies, and all the ones that had these tumors apparently had been behaving strangely.
Right.
So
severe neurological signs.
Yeah. Or lying unconscious or approaching humans and that sort of thing. And when you look at this very gross pathology slide in Figure 1, it's pretty obvious why that would be happening.
Should we make that our show
I think
image?
that'd be a good show illustration.
Yeah. Wow.
Very dramatic.
So the tumor is making them behave oddly, obviously, right?
Yeah, it's displacing the whole brain.
So they say raccoons in the affected geographic region have a nomadic lifestyle and use sewer and water lines for travel and presumed daily exposure to urine, garbage, environmental toxins, and pathogens, including polyomas, which have been consistently detected in raw urban sewage samples worldwide. So maybe they're thinking that a human polyoma was in the sewage and it infected these animals, right?
Mm-hmm.
Yeah, but this is an odd cluster. Or something like that to be happening. It suggests maybe these raccoons have gotten into something novel.
Northern California. I guess you should go out there and figure out what's in this area. Look in the sewers and so forth. So.
Yeah. The question is, what are you looking for?
Right.
Are you looking for the host, the intermediate host, in which case could be anything.
Well, I'd certainly go in the sewers where these animals commute daily. You see these animals getting up and walking through a sewer to work.
Carrying a lunchbox.
I think I'd send somebody else into the sewers to take this
sample and see if you find the virus in the sewage or something related to it, right?
Yeah.
If you're interested in figuring out, I mean, this is a little outbreak and it's never been reported before, so I don't know how extensive. I mean, if it ever happened before or will ever happen again, who knows, right?
Or if they have bats in any of these drain pipes.
I don't think bats fly through drain pipes, do they?
Might roost at the end of one.
Yeah,
I don't know.
Maybe. We'll get a letter, I'm sure.
Yes.
There's a really interesting statistic right in the introduction where they say, during the same period where they diagnosed these 10 cases of olfactory tract frontal lobe brain tumors, no other raccoon tumors were reported in diagnostic laboratories across the United States and Canada.
Right.
So it's just a very amazing cluster.
Yes, so these raccoons come into these labs throughout the U.S., right?
Right.
That's the idea, and so this is unusual, yeah.
Couple things I just wanted to mention about the genome sequence. One is that, like the Merkel cell polyomavirus and the gorilla polyomavirus, there's no agnoprotein, but SV40 has one, and SV40, the agnoprotein is evidently found only in primate polyomas, but it's not found in all primate polyomas. So that I was thinking at first that might give you a clue about the natural host for this virus, but it doesn't really. And then the second thing is, I just found it so weird that they don't have a stop codon that should make the small t antigen be like the other polyoma small t antigens.
Yeah.
So the small t antigen ordinarily interacts with a tumor suppressor like protein PP2A, and that can be involved in the oncogenesis. However, for the Merkel cell polyomavirus, I think it's Pat Moore's work that's shown that you can alter the interaction with PP2A and still get transformation. So, but it's just weird. In two of the 10 sequences, they got a deletion in the intron where you would expect to find a stop codon for the small t, creating the small t antigen protein. And in fact, in those two, it's kind of like the frame shift produces the stop codon, but neither one of the stop codons, they're in two different places. So I just was reading this thinking, how are all these, where is the small t? How does the small t happen? And they speculate on that a little bit, but I just found it weird.
But they have to make small t, right?
Well, maybe they don't. Maybe that's, I don't know. It's just the strange things about this virus.
So what's Mike Imperiali's overall reaction to this?
Oh, just that it was interesting, and the sparsity of large T antigen staining reminded him of the sparseness of T antigen staining in some of the mesotheliomas and other polyoma associated type brain tumors. So he was interested. So.
Well, it'd be interesting to see what they do next, if they do some animal studies or if anyone else picks this up, or if they go out into the environment. Let's see. I'm looking here to see if Eric had anything to say about that.
Yeah, and as they point out,
he said sorry, go ahead.
Raccoons being in constant contact with humans, they're possibly a good sentinel species. You can look in raccoons, and you might see signs of something that's that you might need to worry about.
We sent this great picture of a raccoon with his teeth bared. He said, causality not yet proven, but an association looks good. I don't think people usually think of brain tumors as something that could be contagious, but unlike raccoons, most of us don't go around sticking our nose everywhere. It'll be interesting to see if this disease and virus spreads to the rest of the country. Raccoons certainly get a lot of attention due to rabies surveillance, so we should know more soon. That's from Eric Delwart. That's cool.
Mm-hmm.
All right.
Yeah.
I'm glad I looked that up. Mm-hmm. All right. So that's our 2 papers. Let's do some email. The first one is from Kelly.
Oh, just before you do that,
Yes.
I just saw this in Patty's little note here.
Yep.
This journal, I think, should be totally open access. It's a CDC
Yes,
government
it
journal.
is.
Absolutely.
Yeah,
So-
you just click the link and there you are reading the whole full text.
Yeah, that's really
Beautiful.
nice. It's really good about EID, emerging infectious diseases.
I did notice. I usually, well, I was downloading the PDF for the science paper, so I thought I would download the PDF for this one too, and the PDF for it is not as readable as PDFs normally are. So I
Yeah.
suggest reading this one on the website.
Yeah, this is a weird PDF because I can't highlight anything. Most PDFs
Yeah.
you can highlight.
Yeah, it's all graphic. And by the way, the new highlighting feature in Mountain Lion's version of Preview is awful.
That's
Just
what
thought
I
I'd
use
say.
all the time. But
I use it all the time too, but now that in Mountain Lion they've got it set so that it looks like a scribbled highlighter pen instead of a nice neat computer highlight, which I prefer.
it may be a preference because mine is okay.
I can't find a preference for it.
I'm running
but
Mountain
I'll
Lion.
look some more. Anyway,
Okay, the first email is
That's
from
irrelevant.
Kelly. From Kelly. It's okay, you can rant. Kelly writes, Ni hao, TWiV crew. I graduated 2 years ago from a BSMS program and now work as a technician in an HIV/AIDS research lab in China where the weather is a breezy minus 10 degrees centigrade Celsius and sunny, somewhere above the pollution cloud. Nice. I wanted to contribute to the discussion on the grad student struggling at the bench because her story was nearly my own. In high school, I was diagnosed along with a brother and 3 cousins with ADHD. After years of lost iPhones, countless missing mittens, and the near theft of my boyfriend's car when I left the keys in the door overnight. Psychiatric care and current prescriptions never effectively addressed these issues. Buying cheaper accessories did. Furthermore, ADHD has often been associated with creativity, and indeed I found the medication negatively affected my artistic side, writing and drawing. I decided to go off the treatment. Diagnoses aside, the characteristics described in her letter, good at analytical abilities, solid experimental design, synthesizing and writing papers are aspects of how she thinks. Changing her neurology would also affect these aspects. Of course, associated attention-related errors are detrimental and even dangerous in a lab setting. As a student, I was given similar advice that I was good at writing papers, but possibly not cut out for lab work. Then I moved to China. There is no correlate of ADHD here. When I described my disability to my coworkers, they laughed. At first, I found this frustrating and considered it backward, but then, instead of discouraging me, they showed me tricks and practice to prevent attention-related errors. The saying goes, measure twice, cut once. For me, it was measure 5 times, then double-check again. Progress seemed painfully slow and meticulous, and I was assigned to monotonous, low-resource, low-risk technical work. At times, it took a weekly dose of TWiV to remind me what the heck I was doing this for. But their faith in me was encouraging. As I gained experience, I found I built up experimental habits that made execution almost mechanical. It reminded me of my years as a soccer player, where through repeated practice, the body develops muscle memory. The effort and extra prep work was not fun by any standard, but I was encouraged by my PI, who saw that I was working hard and was highly motivated to pursue a career in a biolab. His attitude was due in part to the Chinese approach towards learning that, unlike the U.S., puts less focus on whether students are innately talented or good at certain things. Instead, intelligence is believed to be products of how much individual effort you put into your education. And she provides a link to an excellent NPR episode on this. I do not suggest that anyone struggling is not trying hard enough, but attitudes on whether we are made for science direct our belief in ourselves and our future careers. I think telling students they have a disorder or are not cut out for science would be a great loss to benchwork. New ways of thinking inject new insights and new connections that might otherwise never be found. My first year was a struggle, but the effort has paid off. Although I still often leave my keys in the door, I do extremely well in the lab and do it quickly with few errors. I don't think I could have reached this point enrolled in a PhD program trying to work out how to address these problems while balancing coursework and other pressures. Much of this was due to my decision to take a few years off and having found such a supportive environment. I am applying to programs in the coming fall, and I no longer worry that my alternative thinking style, some in the U.S. call a chemical imbalance, will not hinder my progress. Anyway, thanks so much for the show and the perspective and personal motivation it provides. I wanted to add this advice to others hoping to pursue careers in science. If you want something bad enough and are willing to work long and hard enough, anything is possible. Happy holidays from around the globe. Wow. It's an interesting take, right, Alan?
A couple of things. Different approaches work for different people, and different people have different severities of different conditions, even within a single diagnosis.
Sure, yeah.
So if you have not developed coping skills for something like ADHD earlier in life, then the process of developing those coping skills could be enough to treat it if you have a not so severe case of it, which sounds like it may be the case here. But of course, I'm not in any way qualified to diagnose anything in anybody. That's my wife's job. But my point in response to the earlier letter was, if you believe that you might have some kind of a psychiatric or neurological problem, or if you're having difficulties like these, get a professional opinion on it. And medication is one possible solution. Coping skills are another possible solution, but in some cases, they may not be enough.
Yeah, well, of course, here, this individual decided to not be in a PhD program to focus on getting the lab work done, and the other individual was trying to figure out whether to stay in the program or not. Yeah. So I wonder what happened to the other person that we
I'm hoping we'll hear back at some point.
Let us know. I mean, you did stimulate some interesting response, so.
Yeah.
Mm-hmm.
Alan, can you take the next one?
Sure. Russell writes, TWiV. This is a follow-up to the letter from Fernando in TWiV 111 about the hantavirus outbreak in Yosemite. This is a link to a nice article in Outside describing the outbreak. Was that 111?
No, it can't be 111.
211, maybe.
It's 211, yeah.
Yeah. And links to an article in Outside magazine. As Fernando noted in his letter, the signature tent cabins had double walls, apparently 2 pieces of plywood. The deer mice were happily living in the space between the walls. Also, in contrast to the canvas tents, they allowed food to be stored in these hard-walled tents. This is in response to the bear problem. After they figured out what was going on, they closed the double-walled tents and tried to close all the gaps that allowed the mice to get in. A 1/4 inch was all that was necessary. They reopened the tents, but the mice soon returned and were happily living between the walls again. So they're tearing down the double-walled tents and replacing them with canvas tents. The critters win again. Merry Christmas to all of you. I'm visiting my daughter in Washington State, and we may have a white one. The temperature here is 6 Celsius, 43 Fahrenheit, and rain, of course. And Russ is normally in New Orleans.
Yeah, Russ looks like he works in infectious diseases. Very good. Well...
Yeah, well, at least they don't have the bears nesting in the gaps in the walls.
1/4 inch, 1/4 inch is... let me just quickly take the next one from Jim, who writes, "Hi all, notice that a few of my favorite podcasts are requesting donations this time of year. You guys put on my favorites of all. Do you fund them with donations?" So, Jim, we don't at this point, but we might at some point. But thank you for offering. We appreciate it. And we're glad that you have offered. We also have another one from Jim, the other Jim, this one from Virginia. Maybe they're both from Virginia, who sent the link to a 20-page PDF from Quora about academic research about applying to PhD programs in computer science. I think we talked about this recently, which is why this comes up. All right, Kathy, can you take Jeffries?
Sure. But meanwhile, I was just checking. I think the first time we talked about the Yosemite hantavirus thing was TWiV 199. Okay. So, and then we maybe have followed up a couple times after, but
yeah,
199.
We had a follow. So Fernando was follow-up, yeah. So
yeah. Okay. So now I'm doing Jeffrey. Dr. et al. And able-minded assistants. 2 quick notes. 1. Episode 210. As I understand it, silkworm cocoon threads are long but do not extend the entire cocoon. Chinese silkworms have a much longer thread. Presumably they're bred for that. Indian silkworms have much shorter threads. That is why Indian silk is considered slightly inferior to Chinese silk. 2. Episode 212. I haven't heard this yet, but I saw glowfish mentioned on the webpage. Have you started to expand your podcast empire to include TWiCG, This Week in Children's Games? Glowfish is the famous children's card game, right? Thanks for the great show, Jeffrey. Yes. And it took me a minute, but then I decided he was thinking about Go Fish instead of Glowfish. And I think once he listens to the episode, he'll realize we were talking about fluorescent pets. So, yeah, that was funny.
Charles sent a link to a YouTube video and asked us to comment on this story in the future of using HIV to deliver viral payloads. This is a video about a young girl who had leukemia and was treated with HIV. And this has been done in a number of people now. The science is in the New England Journal. What they do is to engineer a lentivirus vector to deliver antigen receptors to T cells that allow the T cells to recognize a B cell antigen called B19. And these people have B cell tumors. So in the New England Journal paper, it was chronic CLL. And then the T cells now go and kill the tumors. And many of these people are cured by this. Of course, it also kills their normal B cells, so they don't make antibodies. And that's one of the side effects. But it seems to be effective in treating these otherwise difficult tumors. So that's what that is about. And it's still experimental, but looks like a number of patients have been taken care of by that. The next 2 are all links to stories. Robin sent in a link to an article on what some physicists think about the future of technology. It's an interesting exercise where they asked people, what do you think can happen? Do you think people will be able to use flying cars, jetpacks, live for 200 years, et cetera, et cetera? And what the physicists thought is particularly interesting there. And then finally, the last one is from Ricardo, who sent an interesting article from Boing Boing on another correlation between autism and, this time, organic food sales.
Right.
So this guy made a graph showing how the incidence of autism from 1998 to 2007 parallels the sales of organic foods. Again, demonstrating that if you buy organic food, you will get autism. No, sorry. So it's just, they were just again saying, you can't make correlations like this. So
yes,
that's very amusing. There are many of those. I think we talked about a bunch of those recently, right?
Right. I had 3 picks a week or two ago.
That's right. So there you go. Of course, in the discussion section of that Boing Boing article, there is a lively discussion saying, well, it doesn't prove that there's no correlation. You have to investigate it.
Yes.
Yes. Something like that, though. I mean, what?
First, you have to have a hypothesis that you'd be testing that it is in some way meaningful.
Yes. It's a bit of a crazy discussion. All right, let's do some picks of the week. Alan, what do you have for us?
I have one that I stumbled on a while ago, and I don't know if you picked this for TWiP or I don't think it's been picked on a previous TWiV, but it's a blog called Parasite of the Day. And this is a couple of parasitologists who just pop up it's not exactly everyday updates. Every couple of weeks they'll put up a featured parasite and discuss its life cycle and give a nice little brief discussion of it and some pictures, and then usually they'll link to a reference reviewing it. So it's just kind of a cool little quick synopsis and showing this incredible diversity of parasites that are out there.
Yeah, we have mentioned this on TWiP.
Okay.
Susan Perkins is here in New York City at the Museum of Natural History.
Right.
And Tommy Leung is a friend of TWiP. He writes often and helps promote us a lot. And in fact, we did a story on TWiP once about worms that infect crickets and make them jump in the water and get eaten by fish. And he had worked on a similar interaction with clams, not clams, an organism with a shell that lives on a beach. I forget the name of it. Tommy's going to kill me now. Anyway, he had done some work on that, so it's pretty cool. It's a nice site. But we have not picked it on TWiV, that's right. Kathy, what do you have?
So I picked a tour of the International Space Station, about a 25-minute long YouTube by the Commander Sunny Williams, just as she was leaving November 18th. And it's just really cool. She goes around and shows you all kinds of things in the International Space Station, and it raised more questions for me than it answered almost. I wondered, because the whole time her hair is flying out and just wild, and I thought, I wondered, does your hair stay cleaner if it's not pressed up against your scalp? She talks about some of the other bathroom functions, but she doesn't talk about showering and washing, and then she shows where the food is in their kitchen. But I wondered, do they ever sit down and not sit down, but do they ever eat together? Because I think having discussions over meals is kind of a nice social thing. But so anyway, it was just really fun to watch, and she's very enthusiastic about it, and it's nice to see this whole huge space station. She goes all through it and shows you the Soyuz that they're going to go home on and so forth. So well worth a look.
Yeah, they can't use the shuttle anymore, right?
Nope.
Right.
By the way, Tommy Leung's worked on the New Zealand cockle, right? It's an animal with a shell, but it's not a clam, right? It's a cockle. Mm-hmm. And they're infected by trematodes, which alter their motion. The trematodes go into the foot thing, that thing that sticks out. I obviously don't know anything about this, but Tommy has also written to TWiV as well, as I see here. All right, my pick is a book from NASA called Earth as Art. It's a free PDF, and what it is a compilation of images taken by various satellites that are in orbit around the planet, and these sensors measure various things besides light, and so it's kind of an interesting. It's just a series of these images taken by these satellites. They're very beautiful and they're artistic. It says the images are intended for viewing enjoyment rather than scientific interpretation. So that's a little PDF, or there's an iPad app too, but they're pretty
cool,
pretty nice pictures there.
Yeah, I started downloading it before the show, and now I didn't end up looking at it, but. Yeah, I think that'll be really cool to look at those.
Pretty. Yeah, these space pictures are always neat.
Images, yeah.
I have a couple of listener picks. One is from Robin, our friend who had a comment earlier, I believe. Is that right? Yes. Educated by TWiV. His pick is how the corporatocracy controls scientific research. To the Invincibles, Marcia Angel was the associate editor of New England Journal for a couple of decades until the editor stepped down when the owners of the journal, Massachusetts Medical Society, wanted to sell the journal's name for commercial branding. She then became the first female editor-in-chief for an interim year, conditional to finding a new editor. So this is a talk on Econ Talk podcast, Marcia Angel on Big Pharma. The impact of pharmaceutical companies on academic research, clinical trials, and the political process. Angel argues that the large pharmaceutical companies produce little or no innovation and use their political power to exploit consumers and taxpayers. Is that right, Alan?
She represents an extreme view on a spectrum. And she has a history with this issue that I understand where her perspective comes from. Yeah. Part of it was the controversies going on at the New England Journal at the time she was there. Actually, a large part of it comes from that. And she has formulated a particular opinion that I don't fully agree with. Are you there?
I'm here.
Yeah.
Are you okay? Yeah, that I don't fully agree with, but she does raise some interesting points. I would certainly agree that a lot of pharma companies have wielded a lot of political muscle in ways that are not really about innovation. But I would disagree that they've provided little useful benefit.
Yeah.
I think there's an enormous amount of good science that happens at these organizations. So, there's good and there's bad, and she will focus on the bad. And I think that's actually a useful perspective. But just don't assume that that's the one truth in her take.
Well, check it out for yourself and see what you think. Michael sent along some flu jokes in the Onion. So this is CDC has warned that flu season has arrived a month earlier than usual and notes that this year's strain could be particularly troublesome for the elderly. What do you think? They asked 3 people. One guy said, I'm not worried. I got a flu shot 1 year. Someone else says, every year flu season seems to just get longer and more commercialized. And finally, stay out of Texas and avoid the elderly. Done and done. Nice. I got a flu shot 1 year. That's a good one.
A fairly typical attitude, I'm afraid.
And finally, Jim sends us a link to a database of all TED Talks from June 2006 to the present. 1,396 TED Talks with descriptions and a link to download options. Cool. I thought TWiV, TWIP, and TWIM listeners might be interested. Jim from Virginia. Thank you very much, Jim. And today we have a little shorter TWiV for you. That's good, because last week's was 2 hours plus. So you can catch up. You can find TWiV on iTunes and at twiv.tv. And if you like us, go over to iTunes and make a comment or give us a rating. That really helps to keep us visible there. And do send your comments and questions to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks for having me. I learned a lot, and this is always fun.
Yep, always a pleasure. And Alan Dove is at alandove.com, and you can also find him on Twitter. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. Happy New Year, everybody.
Happy New Year.
Happy New Year.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Next week. What do we got next week? Another rich list TWiV, right?
Mm-hmm. That means it's a poor TWiV?
just unenriched.
Unenriched. We could call it the unenriched TWiV. That would be cool.